Yeah, go ahead, Michelle.
Okay, sorry, bear with me. Hello, everybody. I have got the greatest pleasure to introduce Barry Komisaruk. I met Gary, get me. I met Barry many years ago. It's because I'm thinking of Glasgow. I met Barry many years ago in Glasgow, and I was doing a presentation about Barry's research. And Barry walked into the room. And I was terrified. I was early doors psychosexual therapist, and I was absolutely terrified. And he put me so much at ease, came and sat next to me. And I think one of the first things he said to me was, "I hope you've got a good photograph of me." So Barry started out, from what I remember, this is two people started out in zoology. And just now he's working, he does so much research. And the reason I came in touch with his work was about the vagal system and how that can really help spinally injured people, especially when they know about it, feel areas of their body that they don't think they feel, if you will. He's a distinguished professor in the psychology department at Rutgers University, and he does a lot of research in the fMRI scanning and the scanning of people's brains to look at function and availability to our bodies. Excuse me, Barry, if I've not put that, I've put that in very my layman terms. So I'll pass you over to Barry. Thank you.
Well, thank you very much, Michelle, and thank you, Martha Lee, for both of you for the very kind invitation. So it's a pleasure to meet you all virtually. So let me start. As Michelle said, I started out with in animal studies, and at the time I did my postdoc at UCLA in the Brain Research Institute with Sawyer, and we were studying. He was studying in his lab to teach me neurophysiology. He was studying the connection between the brain and the endocrine system, and where the link is at the pituitary. And there was a model system, which is the induction of pseudopregnancy, which is that sterile mating in rats stimulates the vaginal stimulation, of the mating, stimulates the release of hormones that are involved in pregnancy, but they're not pregnant. So we were studying brain activity that controls the release of the hormones. And when I got back to these were in anesthetized rats. When I got back to my own lab at Rutgers, I continued. I was recording activity, neural activity in awake animals, awake rats, and I tried the vaginal stimulation. And to my surprise, as soon as I applied the vaginal stimulation, they went into this lordosis posture, which is the mating stance, and even if they didn't have any hormones. This was very unusual to me. And they, in addition to that, this is the normal lordosis posture, which was a new observation. But in addition to that, the rats became so immobilized during the stimulation that they wouldn't walk away from the probe. I could slide them forward on a table. They're stiff-legged, and I said, if they're so rigid, what happens if I pinch their foot? And normally if you pinch the rat's foot, they pull the foot back, they squeak. But during the vaginal stimulation, if I pinch the foot, they didn't show any response. They didn't withdraw the leg. They didn't squeak. And so that stimulated me to do a number of neurophysiological recordings of the sensory pathway in the rats in the thalamus, and found that their individual neurons responded to that normally respond to pain and touch. When I did the vaginal stimulation, their response to touch continued normally, but their response to the painful stimulus was abolished. It was blocked. This seemed like it was analgesia. And I said, I did a number of studies that confirmed it in the rats. And I said, really, the most scientific answer I can get to this question, whether vaginal stimulation really blocks pain, is by asking women. So that's how I had the opportunity to recruit Beverly Whipple, because she was in New Jersey on the nursing faculty in South Jersey. she said, I invited her to give a seminar, and I was giving a course in human sexuality, and she came and gave a seminar and said that she wanted to get a doctorate. She wanted to get a PhD. She was a nurse. She wanted to get a PhD, and she wanted to learn how to do research, even though she had just finished writing, co-writing the G-Spot book. So I said, you know, I want to see whether vaginal self-stimulation is going to block pain. And so she did that she agreed to be a graduate student at Rutgers and for a doctorate. And she did this for her doctoral dissertation to study whether vaginal and cervical self-stimulation blocks pain in women. So that's what we did. I devised a stimulator with a modified tampon that if the woman held the handle here and twisted it forward, she could stimulate the anterior wall of the vagina, or if she fitted for a diaphragm, and with a Velcro thing to keep it centered over the cervix, that she could apply cervical self-stimulation by pushing against the cervix. It protected the cervix and also centered the stimulator on the cervix. So we measured pain threshold by this device that puts, there's a motor and drives the weight out, and as it goes out, it puts more pressure on the finger, and this is the point. The finger goes here, so it puts pressure on the finger, and we asked the women, tell us when it first starts to hurt. And at that point, we lifted this up and got the force measure. So we could measure the force at which the women said it hurts. And when we summarize that, if they if this is the control levels, if they apply vaginal self-stimulation, just pressure against the anterior wall, we got an increase of over 40% in the pain detection threshold. In other words, it took more than 40% force above the control to when they were applying the pressure, but it sort of died out after 5 minutes of continuous pressure. But if they did the stimulation repeatedly and in a way that they said felt pleasurable, like rhythmical stimulation, then it went up over 80%, and it built after 5 minutes. And then some of the women, 4 of the women of these 10 had orgasms, and then it took more than 100% increase in force before they said it hurt. And at the same time, their touch thresholds, we measured their tactile thresholds, they didn't change. So that was indeed analgesia. Now, I wanted to know what's the nerve from the vagina and cervix that carries, or nerves that carry the pain blocking signal. So this is a schema of the nerves, and the pudendal nerve carries sensation from the clitoris, the pelvic nerve from the vagina and the cervix, and the hypogastric nerve from the cervix and the uterus. By the way, the clitoris has the highest density of nerve endings of any organ in the body, male or female. So, so in rats, I did surgery in which various surgeries in which if I cut the pelvic nerve, we got the strongest inhibition of the response. Of the pain blockage, so that it seemed like that was the main nerve that was responsible for the effect. So I decided in order to test it in women, I decided to study women who have spinal cord injury, complete spinal cord injury, at a level that would block the pelvic nerve. This arrow is for some reason it didn't come out continuous, but the pudendal and pelvic nerves enter at below T9, below T10, whereas the hypogastric nerve enters the spinal cord at T10, 11, and 12. So I wanted to have to ensure that there would be no input from any of the genital structures. I had this highest level. I studied women with the highest level of spinal cord, complete spinal cord injury that would block all the nerve access to the brain, and that was at T9 and above. So we could separate the effects. And when we summarized the data, it was very surprising. This was the big surprise because this was the group, the women who had the highest level of complete spinal cord injury that should have blocked all the access of all the nerves to the brain. They had the highest level of pain blockage, of increase in pain threshold at the fingertips. So how could that be? Because all the pathways were blocked, presumably. And in addition to that, these women who had the complete spinal cord injury at T9 and above, there were 5 women. They said they have no sensation below the or movement voluntary below the level of the injury, but of all things that were left were menstrual cramps, and the curse of menstrual cramps they did have. They also went in the lab. All 5 of the women said that they could feel the stimulator. Not only did they have the analgesia, but they could feel the stimulator. And 3 of the 5 women actually had orgasms from the stimulation. This was a very emotional time in the lab because they said that they weren't aware that they had, the doctors told them their sex life was over. They weren't aware that they had sensation. And this was the first time they were feeling, they were feeling the vaginal and cervical stimulation. We did these studies in collaboration with Beverly Whipple and also Mitch Tepper. He was a graduate student at the time, and he continued studying the trajectory of the response, the social response of the women as a trajectory of over time after their injury. So that became part of Mitch's doctoral dissertation. So I said, what could be the mechanism for this pain blockage? In able-bodied women, the stimulation at the fingers enters the spinal cord at cervical level and is perceived. When they apply vaginal or cervical self-stimulation, the nerves activate a descending system. What I'll talk about later, descending pain blocking system that blocks the input at the spinal cord level. So I hypothesized that in the women who have the complete spinal cord injury, that the vaginal and cervical self-stimulation would activate the vagus nerve. That's the only possible other pathway. But there was only one study in rats that was evidence that the vagus carried sensation from the uterus and cervix. So I said, well, maybe that's what's happening in the women. The only way we could test that is to look at brain activity in the sensory projection zone of the vagus nerve, which is the solitary nucleus in the medulla oblongata of the brain, the lowest part of the brainstem. That's how I got involved in functional MRI. And what we found was that in all of the 5 women with that level of spinal cord injury, it did activate the solitary nucleus. The kind of primitive fMRI at the time, but we did get activation of the solitary nucleus, which was evidence that it is in fact the vagus nerve that's carrying the sensation. So this got me interested in the projection of other the genital sensory nerves, and we embarked on a study of the projections. So they projected this is the sensory cortex, the somatosensory cortex. This is Penfield's map, the so-called homunculus, where he was a neurosurgeon at McGill University and in Montreal, and he in preparation for doing surgery for epilepsy, removing parts of the epileptic focus in the brain, where he used a handheld electrode and stimulated different parts of the postcentral gyrus. And what he found was that in the wake patients before the surgery, it was with local anesthetic. It was really a heroic study because he did local anesthetic of the scalp and of the skull, removed the scalp and skull, exposed the awake brain while the people were awake and able to talk, And then he stimulated the different parts of the sensory cortex. And here, if he stimulated over here, the people said, it feels like you're touching my face. And over here, they said it was almost exclusively men that he did this with. Over here, if he stimulated a little bit higher up, they said it feels like you're touching my hand. And so he mapped the whole body surface on the sensory cortex and called it the little person or homunculus. It was, what he found was that when he stimulated down in the midline, if he stimulated the more shallow, the person said it felt like he was touching their feet, and a little bit deeper, the men said it felt like he was touching their genitals. So this was the map. And so for our study of mapping this had not been done in women. So when we did the study, here is the, this region of the sensory cortex is called the paracentral lobule. So this top view, the somatosensory, the postcentral gyrus and somatosensory cortex is here. It stops here, and this becomes the paracentral lobule. This is in cross section and sagittal section. This is where it is. For reference, you'll see where our responses are. So this is where he found he mapped the genitals in the paracentral lobule. And when we did the functional MRI, this was in men, we got exactly the same place. This is the corresponding functional MRI. And actually, we replicated, we did a lot of the different parts of the body. We replicated Penfield's homunculus almost perfectly with the fMRI. We got almost the same results throughout the brain, throughout the sensory cortex. So here is the results of studying 11 women who did clitoral, cervical, or vaginal self-stimulation. This is their hand. They're all applying the probe to their using their hands. So this is a hand extended hand area, but this is the region of the where we got the responses to the genital self-stimulation. And notice that they cluster in the paracentral lobule, the same region as in the men. But they're slightly different. Now that probably corresponds to the fact that the clitoris gets input from the pudendal nerve, the vagina from the pelvic, and the cervix from the pelvic and hypogastric nerve. So it probably represents the differential innervation from those three nerves. Now, Beverly had characterized in her book. She characterized the types of orgasms that were elicited by clitoral or vaginal stimulation. And she said that they found that the women described clitoral stimulation or perigenital skin stimulation as localized, and vaginal stimulation-induced orgasms as deep and more heaving whole body involvement, and a combination of those as mixed orgasms. Now, the clitoris is external, and that's somatic. The pudendal nerve is a somatic nerve, whereas the vagina is a visceral nerve, the pelvic nerve, and that could account for a more internal sensation. And now, more recently, we have gotten reports from women, actually from Olivia Bryant in Australia, and some women have told me that the cervical stimulation-induced orgasms feel different. One woman described it as a shower of the stars. Other women from Olivia's study use these very kind of cosmic terms with cervical self-stimulation. And so now if it's via the vagus nerve, it means that it's even more internal. And, at least with in the external, we have there's eye-hand coordination. We have more feedback on what's happening with the external body than the internal body. And that could be the basis for the different types of of orgasms. But the idea is that since each of these different regions can elicit orgasms separately, and you see that the total amount of neural tissue is activated is more when all the regions are stimulated concurrently, that would imply that the more neurons that can be activated by the multiple inputs, the more complex, the more intense and more pleasurable would be the orgasm. In addition, well, here, this is further evidence that women report that cervical stimulation does elicit, is capable of eliciting orgasm. So we changed the map to include the vagus nerve as a nerve that could carry sensation from these regions. And it's very interesting that the cervix, actually, since it can carry, it receives innervation from the vagus and the hypogastric and the pelvic. This is a rare occurrence. There are very few organs in the body that are triply innervated. So it must be doing important things, the cervix. But it's very paradoxical that, and unfortunate that in the book by, the famous book, the Kinsey Report, 1953, the sexual response of the human female, what they concluded was, and what they stated, and this got into the medical literature and the popular literature, that the surface of the cervix is the most completely insensitive part of the female genital anatomy. This was in 1953, and this has become a popular belief. But the strange thing is that this is data from the same book. And if you see, they studied tactile response and also pressure response. And in more than 800 women, and the tactile stimulation of the vagina and cervix was with a cotton wisp, like a Q-tip, but the pressure was with a probe. And here is the data, and I'll show a blow up of it, that the data, their own data, says that when they applied pressure, more than 80% of more than 800 women could feel the cervix and the vagina. So why they concluded that the cervix is insensitive, that got into the medical literature, and doctors believe, a lot of gynecologists believe that the cervix is insensitive. This is the so-called LEAP procedure that's done for where it scoops out using a hot wire. It's really electric cautery. They scoop out, I mean, if there are some cancerous cells in the cervix, they scoop out a large region of the cervix. And what I'm collaborating with Erwin Goldstein who directs the sexual medicine clinic in San Diego. We're finding a lot of his patients complain that after this procedure, they lose a lot of genital sensation. This is one of his patients who is also an artist, and she described these as the erotogenic regions of her body. And after the LEAP procedure, she said it was, it all disappeared. So it's possible, and we see a number of women who have said the same thing, that there's a serious deleterious effect on their sexuality, on their sexual sensations after removal of the scooping out this section using the electrosurgery. It's possible that there's degeneration of all the nerves going to the genital sensory cortex as a result of this very traumatic procedure of burning it, and possibly less severe surgical procedures could be less detrimental. And also, this is an innovation of here's the vagina, the uterus. It's a pregnant uterus. So it's very large. So the bladder, the uterus, the vagina, and the rectum. but it shows the nerves clearly that, there are reports that the Finnish group that a total hysterectomy, which removes the uterus and the cervix, a total hysterectomy is much more detrimental to orgasms than a subtotal hysterectomy. And you can see that in a total hysterectomy, all these nerves, the input from all these nerves will be damaged. Whereas with a subtotal, here, much fewer of the nerve innervation will be involved. A lot of, we reviewed the consequence of hysterectomy on sexual response and found that, you know, after the hysterectomy, many women say that you know, the fact that the pain and the bleeding is gone, it doesn't affect them. It may be that the clitoral self-stimulation induced orgasms is not affected by this procedure, but if they prefer vaginal or cervical stimulation for their sexual response, that would be much more deleteriously affected. And we found in almost all the 22 studies we reviewed for that study of hysterectomy, some women, there were reports that some of the women said that their sexual response was significantly attenuated after the hysterectomy. It's an open question as to whether those are the women who preferred vaginal stimulation. They were never asked. So that's a open question, but again, it implies that there is significant response from the vagina and cervix that the medical profession does not fully appreciate. So we've published a couple of comments on that this year on the importance of cervical sensitivity. And one of the surprising things that we found, completely unexpected, was because as one of the controls, we found that when we asked the women to stimulate other parts of the body, also the nipple, we found that the nipple stimulation activated the same area as of the genital sensory cortex. We expected it to be in the thoracic region, which is far more lateral, but we got activation in the genital sensory cortex. So we analyzed concurrently, and we found that indeed, you know, when some of the women did the nipple self-stimulation, they did get activation in the thoracic region, but that's the white. The white region is the region activated by nipple self-stimulation. But we found that it overlapped with the genital sensory regions. So that was really surprising to all my male neuroscience colleagues, but my female neuroscience said that my female neuroscience colleagues said, yeah, it's not surprising to them that nipple stimulation is erotic, feels erotic. So it's possible that the reason that women say that nipple stimulation feels erotic is that it actually activates the other parts of the parts of the paracentral lobule that other kinds of stimulation also feel erotogenic. There are reports in the literature that nipple stimulation can actually produce orgasms. So it's probably activating, activating this region of the paracentral lobule. So one of the things we wanted to see was what happens if we stimulate the vagus, and there's access to there's an auricular branch of the vagus nerve that innervates the sympa conchae of the ear. And with Eleni Frangos, my doctoral student, we stimulated the sympa conchae of the ear with these electrodes electrically, and the control was stimulating the earlobe. And what we found was that one of the regions activated by the vagal stimulation was, in fact, the paracentral lobule. and also the nucleus accumbens, which I'll show you, that is also activated during orgasm. So this is possibly the mechanism by which the women with the complete spinal cord injury, who had responses to vagal stimulation, to the cervical stimulation, that this is the basis for why they could feel the cervical and vaginal stimulation, because the vagus nerve is apparently capable of activating the paracentral lobule. All these studies got me interested in studying orgasm per se, and to keep the head still during the studies, I devised this kind of severe device, which it's a tracheostomy collar that we first mount on, it goes all the way up to the back of the head. So this makes the head somewhat rigid. And then using the thermoplastic mesh for radiographic studies, molded to the back of the head and another one to the front of the head, cut out holes for the eyes and nose, and make a bite bar. And then this is bolted down to the scanner table. People say that it doesn't feel uncomfortable. It's molded to the head. It's not uncomfortable. I've done, this is me. I've done it myself, and that restricts the head movement to under 2 millimeters, even during orgasm. So here is some of the data that we have for observing what happens in the brain during orgasm. Actually, we had orgasm in some of the women with, the 3 of the women with the complete spinal cord injury had orgasms. And this was actually, we published this in 2004. This was actually the first evidence, first world evidence of where in women's brain orgasm occurs. Nobody had been able to show that before. So here is kind of a time-lapse study in which we use the hot metal analog, where dark red is the lowest relative activity, lighter red is more active, orange is more, yellow is higher, and white is hot. It's like white hot is the highest activity. And we separated the brain. There's a classic division of the brain called the Brodmann areas, based on the different structures in the brain, neural structures, the neuronal patterns. So we use those, and we analyze the data in those terms. And what you can see is this is clitoral self-stimulation. This is one woman, clitoral self-stimulation starting here. And you see that there's activation in the sensory cortex. And gradually the activity increases. And at orgasm, there's highest activity throughout the brain. And then after the orgasm, it cools down. So this is one case. I created this tapestry display where here are the 80 Brodmann regions. Each column is a different region. There are 80 columns, and the whole thing is each row is 2 seconds of scan. We scan the whole brain with the functional MRI. We scan the whole brain within 2 seconds, and these are sequential 2-second epochs, and the total is 5 minutes. So this is one woman where the self-stimulation starts here, and what you can see is there's gradually changing activity throughout the brain, and it gradually increases, and it's ups and downs. But at orgasm, that most of the brain regions are activated, and then after orgasm it cools down. We see this here are 6 different women showing basically the same pattern of the highest activity throughout the brain at orgasm. And this is when they apply the clitoral self-stimulation, the green, different amounts of time, and the red is their orgasm, the times of their orgasm. But you can see that throughout the brain, there's this characteristic highest activity. This is another way of looking at different regions of the brain, showing that at orgasm, comparing the orgasm to earlier stimulation, the greatest activity throughout the brain is at orgasm, and then after the orgasm, it gradually cools down. This is another way of looking at it. We ask the women to press a button at the moment that they say their orgasm starts, and then we can see what you can see is that this is 10 seconds and 10 seconds before. You can see that there's a gradual increase in activity, high arousal, and then at orgasm, many of the regions increase even above the arousal. So we wanted to see what is specific, what is the brain activity that's more specific to orgasm. So we took the 10 seconds after they pressed the button, and this is in 11 women, 10 seconds after they pressed the button, and we subtracted the high arousal in the 10 seconds immediately before, we subtracted electronically immediately before they pressed the button. So that gives us, subtracting out the high arousal, we could see what was different about the orgasm. And this is so I'll show you a summary of some of the brain regions. We try to relate the brain regions that are activated to the various effects of orgasm. Increase in heart rate, increase in blood pressure, the analgesia. We know that the pupils dilate at orgasm. We studied that, we measured that. Other people have shown that oxytocin is released maximally at orgasm. There's evidence from animal studies that dopamine is released during mating stimulation, and dopaminergic drugs potentiate orgasm in humans, and dopamine blockers have an inhibitory effect on it, on orgasm. And also there are orgasmic seizures. These are things that happen at orgasm. And so we analyzed what happens in different parts of the brain in relation to, can we account for any of these functions by what's being activated at orgasm? We see that the insula and the anterior cingulate cortex are two of the regions activated. And it's interesting that these two regions are classically activated by pain. They're activated by pain, but they're also activated by orgasm. So it's possible that there's an inhibitory interaction between in these regions, which accounts for the pain blockage. But you can't tell the active inhibition from active excitation using functional MRI. So we have to do some further studies to try to figure out why there's this common area of orgasm and pain blockage. That's an intriguing and still open question. But one of the things that's known is that the main pain pathway through the spinal cord, the spinal thalamic tract, if it's cut, not only is pain blocked from pain like in cancer pain, when it's surgically transected, the spinal thalamic pathway, not only is the pain blocked, but also the ability to experience orgasms is blocked. So there's a very intriguing commonality between pain and orgasm that we're trying to figure out. Another interesting phenomenon is the pupil dilatation and oxytocin release. We see activation of the paraventricular nucleus region of, this is the hypothalamus. This is where the oxytocin producing neurons are located, in the paraventricular nucleus of the hypothalamus. And it's known that, the Ferguson reflex is a childbirth reflex, that when the head of the fetus pushes against the cervix, it activates a sensory pathway, probably spinal thalamic pathway through the spinal cord, that activates the paraventricular neurons that release oxytocin into the blood, and this that stimulates contractions of the uterus that help propel the fetus out in childbirth. But it activates this region of the brain. So birth canal stimulation, so vaginal and cervical stimulation, activate the paraventricular neurons, and we see this region activated during orgasm in response to vaginal and cervical and even clitoral stimulation. So that is consistent with the Ferguson reflex. Also, it's also interesting that there's convergence from the nipple stimulation and suckling, that this also activates the same neurons in the paraventricular nucleus, that the oxytocin stimulates myoepithelial cells in the mammary gland, in the lactating gland, that squeezes the milk out into the baby's mouth. So there's convergence between nipple stimulation and vaginal and cervical stimulation onto these oxytocin-producing neurons. This is a study by Carmichael and others showing that the peak at orgasm, this is orgasm, there's a peak of oxytocin release into the blood. But now one of the interesting things that we found, so here's a pupil, this is in rats, you can see the pupil dilation in response to vaginal stimulation. And we get this with mating stimulation or with vaginal stimulation, this big pupil dilatation. So what we did a study with Tom Insall at the NIH, in which we studied vaginal stimulation in rats, and we found a release of oxytocin into the bloodstream from the posterior pituitary and into the spinal cord. We did superfusions of the spinal cord. We got oxytocin release in response to vaginal stimulation. Then we inject, this is with Giorgio Sansone's doctoral thesis. We injected, here we show that vaginal stimulation immediately produced pupil dilation. As soon as you apply the stimulation, the pupils dilate. We injected oxytocin directly to the spinal cord, and we got the pupil dilatation almost immediately. And then we gave a oxytocin receptor blocker, and that significantly attenuated the ability of the vaginal stimulation to produce the pupil dilatation. So that, in other words, this confirms this pathway where the vaginal stimulation releases oxytocin in the spinal cord as well as in the blood. But the point is that so oxytocin, this is evidence of oxytocin as a neurotransmitter, not only a hormone but a neurotransmitter, and it stimulates the sympathetic system, which the sympathetic neurons originate in the spinal cord, and they dilate the pupil. So the fact that it's been shown that these paraventricular neurons, some of them bifurcate and go not only to the posterior pituitary where they release oxytocin into the bloodstream, but they also go into the central nervous system, not only the spinal cord, but also the brain. Swanson showed that these neurons bifurcate and they distribute to various parts in the brain as well as the spinal cord. So this is, and since the nipple stimulation can activate oxytocin, it means that nipple stimulation can activate oxytocin throughout the brain. And this is released during orgasm. So this could be an alternative pathway to activate the pleasure-inducing effects of oxytocin in the brain using nipple stimulation. We also see that the posterior hypothalamus is activated during orgasm, and that could account for, this is a sympathetic stimulating area that could account for the tachycardia and the hypertension. And we also see that the nucleus accumbens is activated, and the nucleus accumbens is this very reliably activated during orgasm. And this is interesting because it's been called the pleasure center of the brain, based on studies originally by James Olds, but now by others. And it's been shown that dopamine is released into the nucleus accumbens by genital stimulation, and also other addictive drugs like cocaine and nicotine, also caffeine, and even chocolate has been shown to activate the nucleus accumbens. So it's activated in pleasurable under pleasurable circumstances. There's a dopamine pathway this is the dopamine pathway originating from the ventral tegmentum the midbrain and projecting to the nucleus accumbens here. This is the dopamine pathway. And we see also that the ventral tegmental area is activated during orgasm. So that's consistent with activation of the dopamine pathway. We also see that the dorsal raphe is activated, and the dorsal raphe is a classical pathway for the blockage of pain. The periaqueductal gray also is activated at orgasm. This is the Bass-Baumfield's mechanism of pain blockage, the endogenous analgesia producing system, where the periaqueductal gray is activated by various factors, opiates, but also exercise and even pain. It activates the raphe, the dorsal raphe system that descends, releasing serotonin into the spinal cord that blocks the incoming pain signals. This is the Bass-Baumfield's endogenous pain blocking system. So we see activation of periaqueductal gray and raphe during orgasm, and that could account for the analgesia producing effect. There also, we see activation of hippocampus and amygdala, and these are two areas that are typically activated during seizures. And there are many reports of seizures, of epileptic seizures, feeling orgasmic. There's a more recent study of a woman with epilepsy. The epileptic focus was in the insula, which we also see is activated during orgasm. And what they did was, this woman had epileptic seizures, and she said that if they felt orgasmic when she had the seizures. She had definite genital orgasmic feelings. What they did was they analyzed the connectivity of the different parts of the brain. By connectivity, these are lower threshold regions measured not during orgasm, but just during resting state. So this woman, this epileptic woman, had a hypersensitivity of these regions. And what we found, this is a more recent study we did with Nan Wise for her doctoral dissertation, that these are regions of the cortex that are activated during orgasm. We see that each of these regions is actually activated during normal able-bodied women's orgasms. So this could account for why, when she's having a seizure, then these regions are hyperactivated, and that's why she feels that these seizures are orgasmic, because these are regions that are normally activated during orgasm. One of the strange controls that we did was we said, don't just do the stimulation, just think about it. I thought that this would be, kind of a negative control. But to our surprise, we found that when they just thought of stimulating the clitoris, vagina, or nipple, we got activation in the paracentral lobule, the exactly same genital sensory cortex. And this is a study by Nan, she did part of her dissertation, where she compared physical clitoral or nipple self-stimulation to imagined stimulation. She got activation in the paracentral lobule from both these conditions. But the big difference was that when they imagined the stimulation, the prefrontal cortex was much more highly activated than when they actually did the physical stimulation. So this could be the difference, that the cognitive aspect is much more involved during the cognitive thought of the cognitive process of thinking of genital stimulation than during the actual performance of the stimulation. This is consistent with an earlier study that we did with Beverly Whipple and Gina Ogden. Gina said that she has patients who said they could think themselves to orgasm. And we measured these different measures of pain thresholds, heart rate, and blood pressure, and found that indeed they could think themselves, they could have orgasms. This is objective evidence that they had orgasms. They did the imagery-induced orgasms first, and then the physical clitoral self-stimulation, and the results were significant for both types of stimuli. We also have done some preliminary work on brain regions in some of these women, and we get activation of the typical regions. We also started doing stimulation in men, and we have mapped. So we mapped the different parts of the genital stimulation, self-stimulation in men, also getting activation in all from different stimuli regions in all the paracentral lobule regions. This is, I think, interesting because we asked. I have a device. I made this device to stimulate the rectum and the prostate. The men twist this forward and into the rectum, and then stimulate the prostate more anteriorly. So electronically, I subtracted the activation when they did the prostate and rectum stimulation combined, subtracted the rectal activation from that, and got specifically prostate. So here's prostate stimulation, uniquely prostate stimulation, activating the paracentral lobule, which could account for why men with prostate stimulation feel that it can be erotomanic. Now, it's interesting that the prostate contains embryologically, the Mullerian ducts are what form the cervix. And there's a vestige in men of the Mullerian ducts. It's called the prostatic utricle. And the name of another name for the prostatic utricle, which is the derivative of the Mullerian duct, which forms the cervix in women, it's called the vagina masculina. I love that name. So the vagina masculina. And so here is what happened. Here's in women, in the females, and in women, the Mullerian ducts fuse to form the uterus and the cervix and the upper part of the vagina. So if the vagus nerve carries sensation from the cervix, this is the remnant, this is the vestige in the males of the Mullerian ducts. The fused part, this would be this is the vagina masculina. It's embedded in the prostate gland. So if the vagus can carry sensory input from the cervix, then maybe it can also carry sensation from the prostate in men, and maybe even prostate in women and in men. But this might account for why there are a few cases of anecdotal evidence of men who have a complete high spinal cord injury who say that they can't feel anything, any genital activity or any genital sensation or rectal sensation, but they can feel prostate stimulation. So maybe it's via the vagus nerve. So this is a working hypothesis. In addition, we had the men do nipple self-stimulation, and the men also, the nipple self-stimulation in men also activated the paracentral lobule, which could account for why men describe nipple self-stimulation as erotogenic. See, I'm running out of time. I want to get to some pathology. I'm not going to. We have evidence that there's really dual innervation of the penis, which is not appreciated by the medical profession. But this is evidence that there really is the pelvic nerve as well as the pudendal nerve carries sensation from the penis. I can go, if you have questions, I can go into this, but it's why superficial stimulation feels different from deep squeezing stimulation, because this is like the equivalent of the clitoris and versus the vagina and cervix. We did orgasm studies in men, and we find that the regions of activation are more similar than the sex similarities in regions activated by orgasm. The similarities are greater than the differences between men and women. We also see that nucleus accumbens, we subtracted just quotidian pressure stimulation from stimulation that feels erotic, and we got activation of nucleus accumbens and also the prefrontal cortex in men. This is regions that are selectively activated during erotic feelings. So I wanted to go say something about non-genital orgasms, here this is we published, Beverly and I published a review on non-genital induced orgasms. So, and we reviewed the literature. So people with complete spinal cord injury describe orgasms from these different regions. I did this with Marka Sipsky, neck-induced orgasm. Money described dreams. And in personal communication with numerous men and women with spinal cord injury, they say that the hypersensitive skin area near the site of injury, if it's accidentally brushed by clothing or any kind of accidental stimulation, can be very painful. But if it's stimulated in the right way by the right person, it can feel orgasmic. So this is an interesting question of what pathway, what's the pathway for that? This is, again, it's this puzzling combination between pain and orgasm. I'm not going to go through, you have a recording of this. So this is and you can you can refer to the paper, but in able-bodied people, these are all reports of different bodily regions that, when stimulated, can induce orgasms. Even without sensory stimulation, the imagery, the epileptic seizures, cocaine rush, electrical stimulation, thalamic stimulation, septal stimulation, childbirth. Harold called it birthgasms, women describing having orgasms during childbirth, and also dreaming. So there are many types of non-genital induced orgasms. I want to say something about the more recent studies that we're doing on pathology. Studying, so you have about 5 minutes left. This condition called persistent genital arousal disorder, where there are genital sensations that are very distressing. And actually, we know of some women who have committed suicide from them. It's unwelcome, tormenting genital sensation, even generating orgasms that are not welcome. They don't get satisfaction, and they feel, makes it feel worse. So there was no cause known for it, but a psychiatrist called me and said his wife has it. She has a Tarlov cyst, which I didn't know what it was. I did some literature search, found that the Tarlov cysts form on the genital sensory nerves. And so I contacted a support group, asked the women to send me the MRIs of their lower back. And the Tarlov cysts, which I'll show you where they are, in the literature, people who come in for back and leg pain have Tarlov cysts in between 1% and 9% of the population. But of 24 women who sent me their MRIs with this condition, 18 of them had one or more Tarlov cysts, so which is a much higher percentage. And here's where this is the cauda equina. The Tarlov cysts form right at the dorsal root ganglion, and they can push on the nerve fibers and push on the they can abrade on the pelvis. This is we're collaborating with Chol Kim, who's a spine surgeon in San Diego. This is what a Tarlov cyst looked like during his surgery, and it pushes on this is the S2 nerve root. So it produces mechanical and chemical irritation of the nerve root, producing these causes, causing these symptoms. This is what they look like in the MRI. They're fluid filled with cerebrospinal fluid, typically S2. And so the S2 dermatome is the clitoral dermatome, and the S3 We also see them at S3. That's the vaginal dermatome, but it's also a leg dermatome, and that can account for why women also complain of leg pain and buttock pain with the PGAD. The doctors don't understand this, because if women complain of clitoral pain and they do a nerve block by injecting the pudendal nerve, the women say that the clitoris, now it feels numb, but they still have the pain. And there's one drastic case where the doctor removed the clitoris, and now she had no clitoris, but she still had the clitoral pain. And this was very puzzling to him in the paper. But it's clear that it's had nothing to do with the clitoris. It was because the nerve itself was irritated upstream. And so we published this, and then confirmed. It was confirmed by Feigenbaum, who's a spine surgeon, a Tarlow cyst specialist, for other purposes. And he said that 8 of 11 women treated by the surgical treatment for the Tarlow cysts, their symptoms were gone after the surgery. We also see that men can have persistent genital arousal and pain. And this young man had 4 Tarlow cysts, which were alleviated. We also see that herniated intervertebral discs have produced these symptoms. And I'm not going to go through all the details, but annular tears, anything that pushes or irritates the cauda equina can produce these symptoms. The pelvic nerve and the pudendal nerve carry the sensation, but they also, excuse me, into the S2, S4 levels, but it also carries sensation from the back of the leg. So this can account for why there's the commonality of leg pain, buttock pain, foot pain, and genital pain in cases that are not caused by the genitals or the rectum or the bladder or the anus, but it's caused by irritation of the nerve. And so the doctors looking at the end organs say there's nothing wrong with it, and they don't realize that it's really nerve damage that's causing the symptoms. This is showing that the sciatic nerve carrying sensation from the leg goes into S2 and S3. So we have, and this is just concluding that some have numbness, some have pain, some have numbness. So there's I have a hypothesis that there's a natural history of a gradual, if a low level of irritation, Tarlow cysts or herniated discs or annular tears, low level produces awareness. Chronic genital awareness. More intense can produce orgasms. More intense can produce pain, all these different types of pain. And then if it is prolonged, it can be excitotoxic damage to the nerve fibers in the cauda equina that produces the numbness and the anorgasmia, because that's what we see in many men. And evidence of this is that if we ask the men with anorgasmia or numbness and women also, how did it start? They typically say that there was pain initially, and then it became numb. And further evidence of that is that when Chol Kim does a surgery alleviating the disc protrusions or surgically treating the annular tears or the herniated discs or the Tarlow cysts, many of the people start having recovery, even 10 years after the numbness. So it may be reversible damage. So we're continuing to test this working hypothesis. And so finally, we have more questions than answers, and I won't go through that because my time is up, and these are my collaborators. So thank you for your attention. I'll leave it on the question on my still remaining questions. What are the alternative pathways? Do they all go through the genital sensory cortex, or are there alternative pathways? Can we teach it? can we teach people who have anorgasmia for whatever reason or lack of sensation from the genitals? Are there substitutes from nipple stimulation or thinking? Can these be taught, and can we activate these brain regions to produce pleasurable sensation by alternative means? And of course, the hard question is how do neurons, which are just bags of chemicals, produce any sensation? But that's a whole other existential question. So thank you for your attention. And
thank you so much, Barry. You always blow my mind. I always know that I need to watch that so many times to get the information that I need, because there's so much information in there. There's been a lot of questions coming up. There's been a lot of questions coming up regarding our bowel information. I think Mitchell's answered some of those while we've been going along. Thank you. Like I said, for me, that just blows my mind. And I just think it's...
if there are any men who, can confirm or refute, men with spinal cord injury who can confirm or refute the prostate sensitivity, I'd be very, very interested to know. And, here, let me go back to, here's my, my email address at the very beginning. Please contact me. And let's see, how do I get back here? Here's my email address. Please contact me and let me know. Or if you have any questions about anything in the presentation, please contact me, and I'd be happy to follow up.
Thank you so much for that. I mean, I know through talking to a lot of my clients how nipple stimulation has proved just amazing for them. And not knowing about it initially was something that I hadn't, even me personally, as a psychosexual therapist and a paraplegic at T12, I hadn't really taken on board until I started looking at your research. And that was so powerful for me to actually start to really think about body sensation and about nipple sensation and what I got from that. I remember thinking, my eyes are closed. I know my partner's touching my nipples, but actually I am feeling this in my genitals, and it's amazing. And I think for people to actually have that information enables them to connect their conscious thoughts to actually the science that's involved. It's fantastic. Thank you so much. And if anyone has an interest,
it's activating. I mean, it's a good form of referred sensation, because if this nipple stimulation is activating the genital sensory cortex, then it's activating the neurons that are normally activated from genital sensation, from genital stimulation, and it's a confusion. It's a confusion. It's a benevolent confusion, and it's a referral of the activation to the genitals. It makes perfect neurological sense.
Absolutely. Martha, are there any questions, or Mitchell, that you've noticed on there as they've been coming up? I'm scanning through them now that perhaps we could.
I noticed a question from. Can you hear me?
Yes.
Yes.
I noticed a question from George. He asked if there were any transgender or any gender fluid. That's not the word he used, but gender folks that you studied.
Non-binary.
No, I haven't studied any gender fluid people.
And I was thinking, Barry, when I saw that question, and I knew, or I was pretty sure that you didn't. Would you theorize that if somebody went through gender reassignment surgery, things would change in the brain because of the addition of hormones?
Oh, certainly, that's certainly a possibility. We haven't analyzed anything in terms of hormonal effects, but I think there could be a combination of the actual neural input. I mean, I know in the gender reassignment surgery, there's retention of the original nerves. that should not change. But the cognitive aspect of it would be an additional factor, and that's an unknown. How the cognitive influence, because it's clearly a powerful cognitive interaction with the genital sensory region. I mean, we saw that in the thinking, involving the prefrontal cortex and other regions. This somatosensory too, the operculum. I didn't even mention that, but that's another. It's an alternative sensory pathway, an alternative sensory region in the brain. That's a very interesting, more symbolic region that could be differentially affected in fluid, gender fluid people.
Thanks, Barry.
Very interesting question. Really, there's no research done on it, to my knowledge.
Got to get a PhD student to do that next. Thank you.
Sure.
Thank you so much.
Thank you.
Mitchell will be speaking shortly at 2:30 UK time. So we look forward to that. And Martha, people can stay around and chat if they want to, is that right?
Yes, take a break, please.
I'll say if you want, I'm glad I'm not interfering with Mitch's presentation. And how do I get access to that? I'll stay around until he starts, and I want to hear what Mitch has to say. But how do I?
Barry, all you have to do is just hang on. We're in the same room, so during this break, we're going to get my slides up and get ready for me. All you have to do is hang around.
I don't have to do anything. Just stay with this.
Yeah. It's just one link for the whole festival.
Okay, because I don't see you, Mitch.
Oh, you don't? Huh?
No.
You might have to scroll to the right or the left. There's.
I'm clicking. I see my own. Do I? I guess I should stop the share.
Oh,
Yes.
Yeah.
The sharing has been interfering.
Okay. Now
Okay,
I see
anyway,
it.
I'll end the recording.
Okay, and yeah, that's a good idea.